So glad you all could make it. Like, where's our speaker? She sat down. She's gonna just let me talk for her today. No, you would never want that. Oh my gosh, we'll let her, the expert, share about finches I've never ever seen in my life. So, but I wanted to start a little bit about what happened recently at a board meeting, and share that out so it's not too surprising. There'll be an article also in the next newsletter. So I'm just going to read this statement from our board of directors about this. So we made an important decision as a board the last meeting regarding our chapter name. It's been a 2-year journey for our chapter. On January 2024, at a meeting, we had an informal vote to discuss and decide if we even would open this door of conversation about possibly changing our chapter name. As other chapters have done, to remove the name Audubon. 9 of the attendees of that meeting of 12 voted to open the conversation to explore this issue. A year ago, I think it was, I stood right here and I announced that we're forming a special subcommittee to help work on this issue since we would not be able to accomplish this task with just our regular scheduled board meetings. After 9 months, well, I guess it's a year now, articles updated to the membership in the newsletter, focus groups with high school students, college students, and long-term members of this organization. A survey went to all members of the organization. We have over 50 pages of raw data, and the board of directors voted last week, in a vote of 11-1 of those present to change the name of our chapter. This does not change the programs we offer. This does not change our mission. We are dedicated to our time and efforts to this mission. Our status of the chapter will remain as the National Audubon Society chapter. And we did not take this decision lightly. The board truly appreciates all the input, all the voices that were made into this process. We acknowledge the long history, identity, and personal significance this name holds for many of our members. But ultimately, the decision was driven by the desire to be more inclusive, relevant, representative of the organization's mission and communities it serves today and into the future. For some board members, updating the name is an opportunity for growth, outreach, and long-term sustainability. The belief that the name change will engage more people in this organization and be a stronger arm for the growth of membership. So now you're saying, what's the name? That's the next process we're going to be starting on. In fact, we've started on it for putting that process into place. There will be more opportunities for weighing into this decision. And we'll keep you posted on that. So
it's confusing. You said this is still a chapter of National Audubon.
Correct. Yeah, just like the other 50 chapters that have changed their name. Yeah.
But you're eliminating the name Audubon.
Like other chapters have done, yes. Yep, we're going to remove that name. So, okay, so without, if anyone would like to talk about it, I do have to pick my daughter up by 8:30 and my son, so I can stick around a little bit, but there are other board members here that you can also address if you have questions or concerns about that. With that, I welcome Karen up to introduce our speaker. Okay. Thanks.
So I think I said at the end of the January meeting that coincidentally I met our speaker on the way to the Galápagos in 2024. And we struck up a conversation at the gate at O'Hare. I think I ran into you again in Atlanta. And then we stayed at the same hotel in Quito, coincidentally. And we really fell into conversation then about birds and work and like that. So Jen grew up in La Crosse, Wisconsin, along the Mississippi, and received her bachelor's degree in zoology from the University of Wisconsin. And she did a semester abroad through UWSP to New Zealand. After receiving her bachelor's, she went to the University of Utah to pursue her PhD. And began to work on her dissertation in parasite defense mechanisms in pigeon, doves, and starlings. I think that kind of led her to the work she's been doing on the Galápagos Islands on invasive parasite and Darwin's finches. She said she fell in love with those islands, the people, and their biota. She has been working on she did in the past, right? NIH grant. Let's see, I wanted to share this part. She moved with her family to Dartmouth, where she worked until 2019. Oh, and then they moved to DeKalb to be closer to family. She is a National Geographic explorer and has had the opportunity to continue her research in the Galápagos through that partnership. And she's now an associate professor of biology at Northern Illinois University. So I think we're very lucky to have Jen here tonight, and please give her a warm welcome.
I appreciate that a lot. Yeah, it was a very fortuitous meeting. I really enjoyed it, and it was just great to then get an invite to come and talk to you all as well after it. So thank you for that. I appreciate it. Yeah, so I'm excited to talk to you about the work that we do at Northern Illinois related to the Galápagos. Just to get us started.
I'm going to mess with the lights here for a minute.
Okay.
That's probably the best, huh? Something like that.
Good.
All good? And if you can't hear me, just raise your hand. I can talk pretty loud when I need to. So I'm going to start out with what are invasive species. We've heard a lot about them. They're becoming more and more prevalent, more common in everyday society. And that's because as we move around the globe, we move things with us. And so we're constantly introducing species into new places. When they start to have negative effects on those native ecosystems, we consider them to be invasive. Just a couple of examples to get your brain working on invasives. We've got just a few of maybe some of the worst ones that we know of, or some examples at least, including things like the cane toad and the zebra mussels. We've got wild boars. Lots and lots of plants are considered invasive, things like purple lupine. But, I think things got squished a little bit. So in the world of invasive species studies, we're actually kind of particular about what we call an invasive species. So an invasive species has to go through this process of being introduced to a new place, establishing a reproductive population, and then actually spreading beyond that initial point of introduction in order for us to consider it invasive. So it's really only at that last point that we consider, or we would call it, an invasive species. And what we're interested in when we study invasive species is what allows some species to make it through this gauntlet of processes, being able to make it to a new place, to actually adapt to the environment that it's in there, that's introduced to reproduce, and then to spread. What allows some species to do that and others fail at these various stages? And so, sorry about the wording on all of these. We had to compress images because there are so many pictures in this presentation that Susan had to compress it so that we can all actually see it. So lest you think that invasive species maybe aren't a big problem, I typically start out my talk with this rather ominous picture. These are the four horsemen of the apocalypse. So we have death, war, conquest, and famine. And Jared Diamond, who wrote the book Gun, Germs, and Steel, and a number of other pretty fascinating, if not a little bit redundant books, he coined the term, he borrowed this and instead called it the four horsemen of the ecological apocalypse. And so these four things, habitat loss, overexploitation, climate change, and invasive species, these are the four main drivers of global biodiversity loss. So you'll notice invasive species land in that top four, meaning that they really can wreak a lot of havoc on native systems. They often do this by outcompeting native species or displacing native species. They can alter habitat such that native species can no longer thrive in those same conditions. Sometimes they are predators of native species, but they can drive them to extinction, essentially causing lots of issues. So in our lab, what we do is we study the ecological and evolutionary processes that facilitate successful invasive or successful species invasions. It's a giant mouthful. To say essentially what we want to know is in that process of going from introduction to establishment to spread to becoming invasive, what sort of ecological parameters facilitate that? So how are the species interacting with one another? How are they interacting with their environment such that it allows them to be successful at invading? And the evolutionary factors. So what is it about their genetics or their genetic diversity or their ability to rapidly adapt to the environment that allows them to be successful? And in my lab, we've sort of carved out a smaller niche. So we actually have a decent idea with a lot of invasive species as to what these ecological and evolutionary factors are. What we know far less about is parasite invasions. So invasive species that are actually parasitic in nature, do they have to play by the same rules? Do they have the same ecological and evolutionary factors dictating their success as we see with most other invasive species? The reason we study parasites is that they are incredibly common. So this is one of the most successful life history strategies that we know of. There are some estimates that up to 50% of all species on the planet are parasitic in nature. That's because every known organism on the planet has at least one parasite or pathogen that affects it. So that's where that estimate comes from. It's a very loose estimate, though. These are pictures of just some of my favorites, and I'm not trying to gross you out. I'm trying to show you the massive diversity that we see in parasites. So parasites are found in nearly every major branch of organisms. In order to be deemed a parasite, you have to harm a host. You have to cause harm to a host in some capacity, and that then makes that organism parasitic. So again, what we're doing in my lab is trying to understand invasive parasites. So when parasites are introduced to new places, what kind of harm are they causing to hosts, and what's allowing some to be successful when they're introduced into new places and others not? I used to have to make a bigger case for why invasive parasites were such a big deal until COVID happened, right? Then everyone was on board with, oh, okay, I get it. And COVID is an emerging infectious disease. That's what kind of gets coined into a slightly different bank, but it's the same idea. It's this pathogen that basically showed up in a spot, was able to take root and spread globally, in the case of COVID, right, into new populations. What was it about COVID at that time, in that moment, that allowed it to become a global pandemic? In wildlife, we have somewhat fewer studies to pull on than we do from human pathogens and parasites, but this is sort of one of the most famous. So the Hawaiian honey creepers, there are about 41 species and subspecies, and of those, 17 to 25 of them have already been declared extinct, and nearly all of the remainder are endangered in some capacity. There's a multitude of reasons as to why they have declined and have been declared extinct, including things like habitat loss. So some of those other four horsemen, right? Remember the overexploitation, habitat loss, climate change, and invasive species. But the one that I want to draw your attention to is the invasive parasite aspect of that. So when mosquitoes, Culex quincunfasciatus, was introduced to the Hawaiian Islands, it brought with it avian plasmodium, which causes the disease avian malaria. That disease contributed in a significant way to the demise of a number of these honey creeper species. It also led to some incredibly cool adaptations in the few species that have managed to sort of survive and persist. Things like behavioral shifts, where birds are using the altitudinal gradient of the Hawaiian Islands to adjust their behavior and only be active in places where the mosquitoes aren't, or at times when the mosquitoes are not active. So they will actually move up and down relative to dusk and dawn, which is pretty cool to understand. Turns out that island ecosystems are really nice for studying invasive parasites, for studying invasive dynamics in general, because we tend to know a little more about island systems. They tend to be less species diverse overall than mainland areas. So we have a good idea of what species are present on those islands. at least in terms of vertebrates, things that are cute and fluffy. We really know what are cute and fluffy on most islands. That gives us a good starting point for what biodiversity is there. It also makes it far more apparent when something that doesn't belong there suddenly shows up. It tends to be noticed a little bit sooner than with a lot of mainland systems. So in my lab, whoa, now I broke it. All my buttons.
Oh,
You need help.
There we
there
go.
it goes.
All right. Sorry about that. So
She's getting that pop-up.
I may regret that, but I allowed it to do something. See how that goes. All right, so we are also using an island system to study invasive parasites. We chose to use Darwin's finches and an invasive fly called the Avian Vampire Fly. But before I get into this, I have to give you a little bit of the dirty truth. So this is what I ended up doing my PhD on, but I didn't initially join my PhD lab at University of Utah thinking that I was going to get to go to the Galápagos and work on these amazing birds. I was not qualified in any way, shape, or form. I'd never worked on birds prior to that. I joined a lab to work on head lice. I really made my parents proud. Actually, true story, within the first month of being in my graduate program, I was featured in the Salt Lake Tribune because my PI was advertising his new machine to get rid of head lice, and he needed a subject to pretend to get rid of them on. So there's just a picture of me sitting there sad. My parents were like, this is what you're doing now with your life. But yeah, so I went to study head lice, and I was actually studying avian feather lice and looking at them in pigeons and doves and starlings, when a postdoc came into the lab and said, hey, we just discovered this fly in the Galápagos, and it seems to be wreaking havoc, and Darwin's finches are there. You got any grad students who'd be interested in going to do that? And that's when I beat down everybody else in order to join that program and go there. So I was really, really lucky, but it was truly just fortuitous and luck that I got to go do this amazing work on this amazing system. So I don't really have to tell you all. I've learned that many of you have actually been. How many of you have been to the Galápagos? It's actually a decent number, I think, in this group. Cool. At least 3 groups. Galápagos are islands off of the west coast of Ecuador, down in South America. They were made famous, at least for most of us, by Charles Darwin, who visited them on the voyage of the Beagle. He studied Darwin's finches while he was there and the mockingbirds, and it is said to have helped him develop his theory of evolution by natural selection. Although if you know much Darwin history, what you know is that he actually developed the theory largely based on his work with pigeons. Darwin's finches were just like icing on the cake for him. And he was a terrible naturalist when he was there, which we've all made mistakes in the field, but he actually really didn't take good notes on the birds that he collected from the different islands. And he ended up, it was one of his bigger regrets, in that he didn't know where a lot of the birds he got that he collected actually came from. And so he couldn't trace back their various traits to those islands. It was a major regret of his. All right, I put this in here just in case anyone was starting to drift off, and this is a bird group. So I want to test your knowledge on Galápagos birds. Anyone want to see if they can figure out what these birds are? Just shout it out if you know what this is. Would I stump you on the first one?
It's a frigate bird.
Oh, there we go. It's a frigate bird. I make it easier for you in a couple of slides. Whoops, sorry, that's not a bird.
Not a bird.
That's excellent. Excellent. Land iguana. There, I made it easier for you and put the male of the first species in there. So there's the frigate bird.
Blue-footed booby.
Blue-footed booby. I'm not gonna lie, when I took this picture, I was standing about 12 feet away from these birds. I got to go visit an island of Rabida, and there were blue-footed boobies everywhere getting ready to nest and doing their mating dances. And there were a group of tourists who are walking along, that's really cool, blah, blah, blah. I'm weeping I'm weeping. I'm like, this is the coolest thing I've ever seen. I've always wanted to see them do their dance and they're doing it right in front of me. It was incredible. Oh, this one. It makes it easier for you. There you go. Red-footed movie. Guesses? That's the Galapagos mockingbird, who unfortunately has pox. You see those spots on its feet, the pustules on its feet. So it actually has a pox infection.
Oyster catcher.
Oyster catcher. This one is tough. You just get the silhouette, but there's only one raptor species in Galapagos.
It's the Galapagos raptor.
Yeah, the Galapagos. Oh, yeah. All right. That was just to wake you back up. So the Galapagos are famous for having lots of incredible endemic biota, fauna, flora and fauna. They consist of about 14 major islands. And what a lot of folks don't know about the Galapagos is that they actually have 4 islands with permanent human resident populations on them, including Santa Cruz, Isabela, San Cristobal, and Floriana. Santa Cruz has a town on it of Puerto Ayora of close to 30,000 permanent residents. So it's close to the size of Stevens Point, I think, plopped right there in the Galapagos. These islands have no real freshwater resources. There's a couple of freshwater springs on a couple of islands, and that's it. That means that almost all of the food, now the water is filtered there, but food and for a long time, a lot of the water had to come from the mainland on boats and planes. But given that they are so famous, lots of folks want to see them. So there's a huge amount of tourists that visit these islands every year. Anybody want to guess what the number was? How many people visited Galapagos in 2019, which is our sort of pre-pandemic marker? What was the annual number of tourists?
Million.
Good guess.
2 million.
Yeah, I always get someone who goes with 1 million, and then my number sounds so sad. 300,000. But I should plant somebody who's like 5,000. 300,000. Which, given that there's only the one town of 30,000 and that most of these islands are uninhabited, it is actually a lot of people to move through there in a single year. And so with all of those people come all of the things that tourists need, right? Food and hotels and experiences. And so it's not too surprising that as the Galapagos have increased in their popularity, so have the number of invasive species that have started to pop up on these islands. And one of the worst that we have thus far in terms of the avifauna that's found on these islands is the avian vampire fly. So this is a fly that was introduced from mainland South America into the Galapagos. It was first discovered in the nest of Darwin's finches, so it affects all of the Darwin's finch species. It was first discovered in the nest of these Darwin's finches in 1997. Then we went back and looked through insect collections that had been done, so surveys of the islands to document all of the insect species. And the earliest records of it were actually from 1964. They weren't found in collections prior to that date. So that's our earliest estimate of when the fly likely was around and in enough numbers that it could be collected. So this is a relatively recent introduction in the grand scheme of things. But the fly affects all of the Darwin's finch species. And then once we really started looking for it, what we discovered is it actually is found in the nests of all of the land bird species. We do not find it in seabird nests, interestingly, but we have found it in all of the land bird species. So this parasite is a generalist. It doesn't care which host species it's using. It's happy to use them all. And what it does is it lays, the adult fly is non-parasitic. So it's like a house fly, but it lays its eggs in the nest of, I'm just going to say Darwin's finches from now on, but you can assume it's any of these land bird species. Those eggs hatch into larvae. Fly larvae are called maggots. So it hatches into these maggots that progress through 3 different sized instar stages. And in order to progress from each stage to the next, it has to take a blood meal from its avian host. To do that, what it does is it crawls up from the nest material. So they're living free in the nest material. It will crawl up from the nest material, find a nestling, chew through the skin, and lap up the blood and other fluids from that nestling. They will also do it to brooding females on the brood patch if they have access to that. After it moves through those 3 stages, it pupates, in the nest. It makes kind of a frothy cocoon kind of pupa, and in a matter of 2 weeks, it then emerges as an adult fly and can go on to parasitize other nests. So when we first started working on the system, we truly were one of the first research groups to start asking questions about what this parasite was doing in the Galapagos, what effect it was having. So we really started from scratch. So my first question was just, what effect does this fly have on finch nesting success, right? What is it, I should have that just say finch fitness in general, right? Their ability to reproduce. So we set up a relatively simple experiment. We decided to focus on medium ground finches, which are one of the more prevalent finch species in the archipelago. We stayed on one island of Santa Cruz. We collected a group of these, we found a group of these medium ground finch nests, and then we treated half of them with a pesticide and the other half with water. The pesticide is a simple pyrethrin that we can spray inside the nest prior to eggs being laid. And it has a staying power of several weeks, which is the entire duration from egg through to fledging. So I'm not going to discuss the proof of method. You're just going to have to trust me that the pesticide does its job and got rid of all of these parasites within the nest for the entire time that we needed it. The water was used just to make sure that we were treating the control nest in a similar fashion, disrupting them in a similar fashion. Then we let the finches do their thing. So they got to go lay their eggs, have their babies. And what we did is we monitored the nestlings. So that sounds easy, except Darwin's finches are kind of jerks, and they put their nests up in these giant opuntia cacti and like embed them way in there. So our job was to carry around our 10 to 13 foot ladder, crawl up that, reach our hand into these cacti and pull out these little squishy flesh blobs and measure them, right, for growth and other things, trying to not drop them on the ground. I never dropped one, never one. But you can imagine it was a little bit hard. It took me like 5 years after I finished my PhD before the last cactus spines came out of my skin. But what we wanted to do was measure growth and survival of all the nestlings in either the treated or the untreated nests. We also put in these cool bullet cameras. So these are like GoPros, that snowboarders put on their helmets so they can keep track of their tricks and things like that. We use those in the field because you can attach them to a car battery and run video surveillance essentially on these finches to see what sort of defense mechanisms they might have, right? When the adults are coming to the nest, or the nestlings are in the nest, they have any way of actually getting rid of this fly or dealing with it in any capacity. And then finally, what we did is when a nest fledged or failed, meaning all of the nestlings either left the nest or they all died, we would collect the nest from the field. Darwin's finches typically don't reuse their nests. Sometimes they'll build one on top of an old one, but they don't ever reuse the same one. So we could collect them from the field, bring them back to the field station where we were staying, and carefully dissect them, counting out every single parasite we found in that nest. So the larva, the pupa, the closed pupal cases, meaning the empty pupal cases. And what that did is it gave us a really good estimate of how many parasites were in each of those nests, which that's often a very difficult thing to do in parasite studies, is to know just how many parasites are affecting a particular host. So this is a little bit of a blessing within this system in that we could get accurate data like that. All right, this is a simplified graph that represents 5 years of my life. So I like to leave it up here for a long time and explain it to you all. So what I've done is for each year that we did the study, I took all of the nests within one treatment and gave you an average of their mean parasite abundance and then the fledging success of those nests. So how many parasites on average did we find within the water treated nests, and how many of those nests managed to successfully fledge young, versus in our pyrethrin treated nests, how many parasites did we find, and how many fledgings did we get? And so what I'm hoping you can see, if you ignore this point right here for a little bit, is this really nice line where essentially the more parasites they have in the nest, the less likely they are to successfully fledge any young. That's a little bit of a duh moment, right? If they're parasites, we know they're bad. What this lets us do is say how bad they are. Turns out they can be really bad. So we actually had a couple of years where there were absolutely no nests in the entire breeding season that managed to successfully fledge offspring, right? Fledge young, which is not a great result by any means. Even with this particular point in here, we get this significant relationship between these two. This point reflects a year that was extremely dry. And so very few birds nested at all. They will often just forego nesting if they aren't getting cues that there are caterpillars and other invertebrates around to eat. They just won't nest. So the only birds, these were in unparasitized nests, but the only birds that managed to nest were the really like the first year males, right? Basically all the inexperienced parents who were like, we can do this. They couldn't do this. And so they failed anyway. The other thing that I want to point out are the two pictures that I have on each side. So you all are bird people, so this probably doesn't freak you out as much. This is a very healthy looking nestling. When I give this talk to non-bird people, they're like, ew. No, this is a super healthy looking nestling. So this is a 7 or 8 day old nestling. It's nice and pink in color. It's good and plump. Its eyes are open. It has these nice pin feathers. Well, this is an example of a nestling that came from an unparasitized nest. By contrast, this is also a 7 or 8 day old nestling that came from a heavily parasitized nest. You'll notice it's smaller. It's lacking the pink color. That's largely because it's been exsanguinated, so it has lost so much blood. The eye is still closed. The pin feathers are significantly smaller than on this one over here. You may also notice up at the nares, they're enlarged. The first instar larvae occasionally reside in the nares of these birds. The later instar larvae are who left these giant holes in the abdomen of this poor critter. So there was a stark contrast in the health and well-being of these nestlings between parasitized and unparasitized nests.
These are mean parasite abundance, so I'm curious approximately how many data points made up each mean dot?
Oh, that's a great question. It's different for every year, but it was our smallest sample size in our year that really dry year. This is like 6 nests. For all the rest of these, it's over 20.
Okay.
For each one. Yep.
Thank you.
Yep. Sad day. Okay, next we wanted to look at defenses. Do these birds have any defense capabilities against this fly? And one of the things that we expect when we're studying invasive parasites is that these birds don't really have any evolutionary history with this parasite. So we don't expect them necessarily to have any defense mechanisms that they've already evolved. And when we went looking for them, we couldn't find them. So we did not find any behavioral defenses. This is a video that I could not get to play. I apologize, but this is an adult female from inside the nest. I've got a circle around a larva that she essentially picks up, gums a little bit, and then drops right back into the nest. They're taking all kinds of other stuff out of the nest, right? The fecal packets, any other insects that they're finding in the nest, they're cleaning out the nest. These larvae are giving them no cues whatsoever that it is something that needs to be removed from the nest. But what's also surprising is they don't eat them. They're eating caterpillars and other invertebrates, and that's primarily what they're feeding to their nestlings, but they won't eat these larvae. So we think that there may be some sort of chemical cue that's coming from them that says, like, I taste bad, don't eat me, right? And so they're just spitting them back into the nest. We looked for immunological defenses. So might they be mounting antibody-specific immune responses against these parasites that could help them out? The adult females, yes, they do mount an antibody response in after they've been bitten by a parasite. Whether or not that actually improves their fitness is not something that we were able to measure. But what I want to point out is that the nestlings down here had essentially no antibody level response to being bitten by these parasites. And that likely is an ontological thing. So they are born immature with immature immune systems. By the time their immune systems have matured enough that they might be able to start producing these antibodies, they're dead. So it's just the timing doesn't work out for them. And then one of the last things that we measured was actually cortisone. So whether or not they might have an adaptive stress response in some capacity to these flies, we again found no evidence for that. So a pretty bad story in general, right? We've set up that this parasite is bad. They have no defenses against it. What are we going to do about it? So there are a few glimmers of hope. There are things that are happening. We are working with the Charles Darwin Foundation and the Galápagos National Park to try to come up with these management methods, because some of these species of Darwin's finches are considered endangered. There's the critically endangered mangrove finch. There's a lot of interest in trying to preserve this incredibly valuable set of endemic species. So these glimmers of hope. I love showing this with you. So, or this picture. This is a clothesline back at our field station. So we would hang our clothes out there. And what we noticed is that we had a couple of finch visitors every day who would come and they would pull the cotton off of that clothesline and take it back to their nest. We could watch them go back to their nest with it. Often when we were in the field and we would take a nestling and take a blood sample, and we'd put our cotton ball down on the ground in order to finish what we were doing and put that nestling back. When we come back, the cotton ball's gone. Like the females had come in, swooped in and taken it. They would pull our hair out of our heads while we were working on the nestlings to go incorporate into their nest. So they were kind of eager to have this soft stuff. And so my colleague, Sarah Canuti, had this idea, well, why don't we have the birds do the hard work for us? What if we put out cotton that had this pesticide already laden into it, and the birds could come grab it and take it back to their nests? So as part of her PhD, which immediately followed mine, she did a great experiment where she set out cotton that had pesticide on it, cotton that had just water on it and had been left to dry. The birds took everything. They wanted it. All of them that took the pesticide-laden cotton managed to reduce the parasite load in their nest such that they increased the fledging success, which is pretty awesome. So the Galápagos National Park is actually using this method. They are putting out this parasite-laden cotton in particular parts. What we learned is that those mangrove finches, those critically endangered mangrove finches, are a little bougie. So they are a little more particular about what they put in their nests. Cotton was not something they were interested in. So we had to experiment with different fibers, twigs, grasses before we found one that they were willing to take and incorporate into their nests. The other glimmer of hope is that there are now these parasitoid wasps that are seriously being considered for release in the Galápagos. These are parasitoid wasps that infect the Avian Vampire Fly in their native range in South America. What they do is they lay their larvae inside the pupae of the flies. When their larvae develop, they eat the fly pupa and then emerge as a wasp. But they have no effect on the birds. They just kill the fly, which is great. So there have been all kinds of studies to try to understand whether there will be incidental or accidental hosts that they may be infecting in the Galápagos, what the pros and cons of any sort of biological introduction will be of that sort of thing. But it is I think it's extremely likely that it ends up happening at this point. All right, so quickly, I'm just going to run through what we're doing now, what we're doing next. So we identified how bad the fly is. We identified that the birds don't really have any defenses. But one of the things that we can still pursue is trying to understand where the flies came from such that we can study them in their native range and in their invasive range to understand what parameters might be limiting them and limiting their distribution, limiting their population sizes in the native range that are not doing that in the invasive range. It also helps us get at that initial question of what ecological and evolutionary factors allowed them to get to Galápagos and be so successful. So to do this, we have to use a genomics approach, a population genomics approach. So we collected 95 flies. Oops, sorry. Oh, I lost all kinds of images there. Well, this is a map of Ecuador and the Galápagos. We collected 95 flies from across the archipelago. So we had 5 on the islands and 2 in mainland Ecuador. And then what we did is we sequenced the DNA from all of those flies and we described the relatedness of different populations. So when we get a DNA sequence back, essentially we can say when 2 populations have very similar genetic sequences, they're more closely related to one another. Whereas if they have very different genetic sequences, then they're more distantly related to one another. And you can do this within a species, just looking at different populations. And that gives you some idea of potentially what populations sourced other populations. So when we did this and we ran our analyses, what really came back was that of these 7 different populations that we studied, they all grouped into 2 main groupings, the Galápagos Island populations and the mainland populations. This told us 2 different things. One, the mainland population that we sampled from was probably not the source population for the Galápagos. That's not super surprising. We went and sampled 2 spots in Ecuador and that's it. So we're not too surprised we didn't find the source population of the Galápagos, but it does tell us that they're very different. The other thing it tells us is that all of these Galápagos flies from 5 different islands essentially looked genetically identical. That means one of 2 things. I guess they're not mutually exclusive. One, the fly hasn't been in the Galápagos long enough to have accumulated different mutations on the different islands, such that they all still look the same from that initial introduction. Or they're simply moving around the islands, such that it's like one big population. The flies can move between them. So what we did is we paired up with National Geographic and Lindblad. They sent out a thing to all their call to all of their National Geographic explorers and said, hey, we have this boat that goes around the Galápagos showing tourists around, but if a scientist can make use of being on that boat to do research, let us know. Yes, we can. So we went on that boat trying to understand whether or not we could catch the fly in the process of moving between different islands. And we broadened the study out to say what insects in general are potentially using these boats to hitchhike between islands. So we got to go on the National Geographic Endeavour 2, which is one of their bigger boats. It holds about 100, I think it's 100 people in total. So it's like 70 guests and 30 crew. So it's not like a massive cruise ship, but it's decent in size. And we were the weirdo scientists on board who hung up all of these traps all over their nice, beautiful decks, like deck areas, trying to see whether or not we could catch these flies. This is me setting up a trap with a very cheeky fly who decided he was going to sit on the outside of it while I was setting it up. I did not manage to catch him though. What we found, oh yeah, I also got a really fun opportunity while I was on the boat the second time. This is the trip that I met Karen at, actually.
Oh, yeah.
I was able to be featured on Good Morning America because they were on the boat at the same time. It was just by chance on the boat at the same time, featuring the Galápagos and trying to discuss the effects of climate change on the Galápagos. So it was really fun and cool to do. And so I got to talk about the place I love. So we followed the boat as it went on this wild itinerary all over the different islands. And we collected insects every single time that we stopped at one of these next island spots. So we collected all of those insects. We identified them, we preserved them, we identified them. And the major takeaway is that even on these boats that have really high compliance with methods that are meant to prevent the insects from hitchhiking between islands, we still found a ton of insects. So they are absolutely using these boats to move between island places. And relative to this talk today, we found avian vampire flies on the boat. In fact, we found gravid females, meaning that they were like pregnant females that were potentially being moved between the islands. So you really only have to introduce one in order to have a potentially successful introduction. So what that tells us is that, yeah, the Galápagos may truly be considered one population for these flies. So if we're going to treat one island for the flies, we're not going to be very successful. We have to treat all of the islands for the flies if we are going to be able to address the issue. The next thing to come from this is that we realize we're lacking in a more in-depth tool to allow us to really understand the differences that might exist between these populations at such a fine scale. So what we needed to do was create a tool to better understand the native versus invasive population ecologies and a tool to follow how selection is shaping traits among invasive populations within the Galápagos. So again, this means genetics. So what we did is we created a reference genome. So the first study we were able to do using genetics, but relative genetics. So you're comparing one organism's genetics to another organism's genetics. but you're not actually saying, well, this gene that makes this protein is the one that we're going to look at in all of these flies, because you don't know what you're looking at. You're just comparing the whole sequence. What a reference genome lets us do is try to understand which things within that genome are starting to differ and change, which things are important within that reference genome that may have allowed it to be successful. So we sequenced an adult female fly, and then what we did is we compared it to 7 other species of closely related flies. And we said, well, which gene families in the avian vampire fly are being heavily upregulated or downregulated as a clue to what might have helped it be successful. And we found these 13 different gene families that end up being heavily upregulated or downregulated. And the ones that I want to point out, the cytochrome P450s and glutathione transferases are both associated with the development of insecticide resistance, which is not great. It doesn't mean that the flies are resistant to insecticide. That's not what that means. What it tells us, though, is that these flies, in a way, may be more predisposed to be able to tolerate a lot of toxins in their environment or a lot of potentially insecticides in their environment such that they can thrive. So this is really something to sort of pay attention to. We sequenced more flies. Now that we had this reference genome, we sequenced more flies, again, from the 5 different islands in those 2 places in Ecuador, just like we did in our first study. But we used the reference genome to try to highlight specific gene level differences that may be happening or that may be showing up in these populations. And what we found is, just like our first study, our mainland population looks very different than our island populations, genetically speaking. But there's a ton of diversity within the mainland populations that we do not see in our island populations. So again, we had hoped the reference genome might help us pick out these finer scale differences between the island populations of flies. And we really had a hard time seeing that. But what we did do was a phylogenetic tree of all of the flies collected from these different islands relative to the flies collected from the mainland. And what we expected is that we would get a full mixing of colors over here, meaning all of the flies, no matter which island they came from, looked the same. So the tree basically put them all mixed together with their branches, which is for the most part what we find, with the exception of this one grouping here, which is from Marchena Island, which, if you'll notice, is sort of the most removed of all of these islands, right? And here's what's really cool. When we map that to the routes that a lot of these tourist boats take, Marchena is not one of the islands that is regularly visited by tourist groups, which is pretty cool. So it tells us that these boats may really be contributing to the movement of these flies between islands. But it also tells us that when a population becomes isolated, relatively quickly, it may be responding to that local environment and changing its genome, essentially, right? Changing what genes are being favored by selection. The other thing I want to point out on here is the only 2 islands where we have never found, sorry, I keep wanting to call it Flornus, the avian vampire fly are Española and Genovesa, which are on those tourist boat routes. So what we've been able to do is highlight the extreme risk that these boats posed of introducing this fly to those islands. It may be, though, that those islands for some reason are unhospitable to this fly, which would present a very cool case for trying to study what it is about those islands that makes them more resistant potentially to the fly. It could be that it's luck, could be that they actually have some sort of trait. So, oh, I have my picture of there's a very cute picture of a finch right here, just so you know. So here are the major takeaways from this. We have ongoing projects in our lab that use the reference genome to enhance studies of population structure and identify potential source populations. So we are trying to get more samples from South America to try to really understand where this fly potentially came from. We're looking for signatures of local adaptation among island flies. So we're going to go back to those islands of Marchena and the islands that we studied to see if we're starting to, if we can pull out signatures of selection, meaning how the environment is shaping their genome. And our ultimate goals are to better understand those ecological and evolutionary processes that make invasion successful for certain parasites, in particular this fly, and inform potential management strategies by studying the fly's ecology in both its native and invasive range. So with that, lots of folks to acknowledge. None of this work was done alone. Every bit of it was done with a team of workers. We still have collaborations that go on between multiple universities and of course with the Charles Darwin Foundation. And then huge thanks to funding efforts as well. So with that, I'm happy to take any of your questions. Thanks so much.
How big are the larvae, actually?
Yeah.
That you find in the nest, are they like?
Good question. How big are the larvae? So the first instar larvae are very small, just a few millimeters. The third instar larvae are about the length of your thumbnail. They're big, given that the nestlings from most of these finches are, yeah, I would say at just about at fledging, they're about this big, and you've got a thumbnail sized larva going after it.
When the parasitic fly bites one of the nestlings or an adult, I would think the feathers would be too thick to penetrate. So do they bite the legs?
Good question. So they have an easy time biting the nestlings because they're naked little flesh blobs in the nest, right? The females, they're able to get just through the brood patch. So when the females lay down on the nest, they've lost all of those feathers on the brood so they can have direct skin contact with the eggs. That's where they're likely getting bit.
I see.
When they're doing that. We don't really see much on the legs, probably because of the scales make it actually pretty tough. We did have evidence every once in a while that they would get through the padding of the bottom of the foot.
One other question. For those situations where the cotton didn't work, maybe I didn't listen carefully enough. What did you substitute for the cotton?
Oh, good question. So for the mangrove finches that were finicky, I think they're using a combination of, it was like, I can't think of the name of the fabric. You use it when you go in the field, like a really thick, like a canvas, canvas, threads, and grasses is what they ended up using. But even then, the mangrove finches weren't as particular for it. So they actually have teams of researchers that go out and find every single mangrove nest and spray it. They directly spray it.
It looked like these particular finches are seed eaters from the size of the beak.
The medium ground finches are seed eaters, yes.
And did you use other species besides the medium ground finch?
Yeah, so we've now done this with small ground finches. They've done it with.
Now there's a large one too.
There is a large one. The large one is not in super high abundances, so it's a little tricky to study.
Yeah.
And their nests are really elusive. We don't know why. They're really hard to find where they nest. And there's also a decent amount of hybridization that occurs between the large ground finch and the medium ground finch. That makes it tricky as well. But no, they've studied it in a number of the different finch species at this point. We seem to see the same effects in all of them. One of the interesting things, though, is when they looked at the effects of the fly on mockingbird nests, they found that those nestlings were far more tolerant. So they still had decent nesting success, even though they had similar parasite loads to the finches, which is probably a size thing because the mockingbird nestlings are just much bigger than the finch nestlings.
So you found the medium finch, ground finch, even on those two islands, Genovesa and Espanola, that don't have any of the parasitic fly. Did you find those birds there?
They were the medium ground finches on. Oh, now you're testing my knowledge.
I'm
I think
sorry.
they're found on both of the islands. They looked at it in any nest, so they went and collected any and all nests that they could find, and they never found a fly larva
Very good.
in the nest. Yep.
Thank you.
You said that they spray or that the birds build a new nest each time. So when do you do the spraying? Do you wait until there's eggs in it so you know it's a new nest and it's going to be used, or do you spray as soon as you see in a nest? Or
Good. So so the males will build a nest. The females then come and check out that nest and either say, yeah, this is fine. We're going to use this one. Or they say, well, that was cute. Oh, we're gonna build a better one over here now. And we are tracking all of that. So we are going through the field every single day with binocs, watching these pairs of birds and saying, okay, they're actively going in and out of this nest. That means they're likely to use it. And so what we do is we sort of track nest completion, and we try to get in there. You have like a 4-day window between nest completion and egg laying to start, that you get in there and you spray the nest. Every once in a while, we would go to the nest and it would end up having eggs in it already. And what we could do is very carefully remove them, remove just a little bit of the top nesting material, spray the nest, let it dry, put the nest material back, put the eggs back, and let it go. So we did have a few that we actually did in the egg stage, but most of them we got pre-egg. And some of them we treated, and then the birds were like, just kidding, we're gonna build this one over here.
What are you treating? What chemical? Pyrethrum?
Yeah, it's a pyrethrin. Yeah. Just an aqueous solution.
The tour people aren't gonna like you telling them they can't be carrying these flies from island to island to island.
Yes.
That could be a little bit of an issue for you.
Yeah, it's a good point. So we were actually really nervous when we asked National Geographic if we could do this, because we were essentially going to tell them they were messing things up, right? That it was a problem. And so we anticipated that they were not going to want to comply. But to their credit, they truly are science driven and said, well, if it's a problem, we want to know it's a problem so we can work to find a solution. So that was really impressive. And they are exploring other kinds of protocols to continue reducing the number of insects. So they do a number of things, like they have different lighting so that it's less attractive. They have blackout shades that they put down at night to keep interior light from shining out. They have bug zappers on the outside of every deck to draw in insects. And the deck crew works very hard to spray down all of the surfaces whenever possible. The problem is they have a policy that if a guest is present on the deck, you are not to ask them to leave so you can spray down the thing. Whereas we as scientists were like, get them off, like, let's go, we're gonna spray the thing down. So they're interested in trying to review policies like that so that they can say, hey, we can educate our guests and say, this is really important that before we lift anchor, we spray down the entire boat. So hopefully we can get some of those policies in practice. I think, again, the National Geographic boat is probably one of the most compliant. These policies exist, but they are rarely enforced. So there are plenty of boats in the Galapagos that won't or don't understand why it's as important to continue doing that. So it's a long-term effort.
How did you guys house? You're having to check all of these nests.
Yeah.
Did you stay on one island or were you?
Yes, this was on one island. So all of the nesting stuff, all of the tracking of the effects was done on one island and in one area. So we had about a 3 kilometer by 3 kilometer area and a team of 5.
Was it Santa Cruz?
And every day.
It's all the nest watching.
There's a lot of nest watching. Yep.
Was that on Santa Cruz?
That's all on Santa Cruz. Yep. Yeah, we did it in the El Garapatero area, which is about 13 kilometers away from where that main town is. The Charles Darwin Station has done studies every year in town on the flies. And my collaborator, Sarah Canuti, has done some really cool work looking at urban finches versus rural finches and the effects of the flies. And she just recently published a paper that said that some of the urban, essentially living urban, can help the nestlings survive these parasite infections a little bit better, probably because of all of the available extra food. So anytime tourists are leaving food out on tables, the finches are on it instantly, just like house sparrows are here or pigeons are here. It's the same thing there. The Darwin's finches are swooping in and grabbing rice and they're off in no time. But that added food may be what's needed to sort of help their nestlings survive the parasite. It's not complete, it just increases it a little
This is a real easy question,
bit. Good,
real easy.
maybe we can end on that one.
You probably had graduate students help you on this project.
Yes.
Were they masters or PhD students?
Well,
It's not an easy question.
no, the answer is all of the above. We've had masters, PhD, post-bacs, so those are students who have their bachelors but are not yet in grad school, and postdocs all working on it. So it's a full team effort, and it's multiple lab groups all combining efforts in order to do it.
It seemed like when I was working with graduate students as a biology professor, you train quite a bit of training, and
Yes,
sometimes a master's degree, boy, you really invest a lot of time in this student, and all of a sudden they're done. Whereas a PhD student, you were really getting your
yeah.
return on
The
investment.
master students have done most of the lab genomics work. The PhD students are the ones who can be in the field, because of the training time.
Yeah.
Yeah.
Very good. So you offer a PhD at NIU?
Yes, we do. Yep, we have a strong PhD program and a master's program. Yep.
Very good.
So what's the main source of your funding?
For this work, it has been National Geographic. What else have we pulled from recently? I think it's actually been almost exclusively National Geographic. And then a collaborator who did the reference genome had money through Kent State. They had an internal grant that he used to fund that project. All of my other work on my local system comes from NSF. I have a big grant to do that, and then local DNRs help contribute to that.
Is that, any of that in jeopardy given the current situation?
The type of NSF grant that I have is a 5-year grant, and it has been promised to us that it will continue, that it's been designated and set aside. The nice part is I already have 3 years of the money like in hand, so we will not give it back. Whether I get those other 2 years, who knows? The only funding that we've had jeopardized thus far were collaborations through USGS. So USGS kind of imploded and is now back. But as a consequence of that, they lost 25% roughly of their like top tier, not top tier, but their higher level researchers. So a lot of programs are now without leadership. So it's a problem.
Sense.
We gotta end on a happier note than that. Sad one to end. And for all of the grief that I gave about tourists visiting Galapagos, I do highly recommend it. It is an incredible and amazing place. The people are wonderful. You will never see wildlife like you can see there. I mean, some of the species truly are just endemic for there. So I do highly recommend it. Just do it responsibly. And yeah, we'll go with that. So thank you all. I really appreciate it.
Okay, we have snacks over here. I think there's, especially some noodles. It looks like a nice variety of cookies. So coming up next month, we have Terry Dalton, who's going to be with us through Wisconsin Clean Fire, talking about science and public policy. So, see you in March.
I visited Galapagos twice. Once, by the way, since 2003 and then 2024. It looked very different.